Histology is the study of tissues, and for many students it is the most difficult unit in anatomy and physiology. In this unit we're going to be looking at epithelial tissues. So what the questions we'll be asking is, what is the name of the tissue, and how can we identify that tissue using simple yes and no questions? Our first question is, does the tissue have a free surface? A free surface, or free apical surface, of a tissue layer means that the cell layer surrounds an open space, lumen, or body cavity. If the answer is no, then the tissue is either muscle, nervous, or connective tissue. All of these tissues will be addressed in another presentation. There are three types of muscle tissue. This photomicrograph is of cardiac muscle tissue. This is a multipolar neuron. This photomicrograph is of areolar tissue, a connective tissue type. If you answered yes, there is a free surface, then you are observing an epithelial tissue layer. Keep in mind, epithelial layers are avascular. That is, they do not contain blood vessels. They are lining tissues. They are actively mitotic and attached to an underlying tissue by means of the basement membrane. Your next question should be, is the tissue composed of more than one cell layer? If you answer no, then this is a simple layer, which will be further identified by individual cell shape. Remember, simple layers are found in areas of the body where active absorption, secretion, and excretion are important. They are, to a certain extent, protected from environmental forces. Pseudostratified columnar epithelium are found in the trachea and upper respiratory tract. All of the cells are attached to the basement membrane, but not all of the apical surfaces reach the tissue surface level. Because the nuclei are at different levels, the tissue appears to be stratified, but it is not. This is a single layer of cells with different cell heights. The green arrows are pointing to goblet cells. Goblet cells are unicellular glands that produce mucus. Simple cuboidal epithelium is found in kidney tubules, the surface of the ovary, and in some ducts and glands. The cells are as tall as they are wide and appear square in cross-section. The yellow arrows are pointing to kidney tubules. Simple squamous epithelium is found in the lung alveoli and in the glomerulus and Bowman's capsule of the kidney. In cross-section, these cells are very thin. The black arrows are pointing to the nuclei of the simple squamous layer. Simple columnar epithelium is found in the kidney and throughout the digestive tract. The columnar cells are oblong or taller than they are wide. The nuclei usually all appear in a straight line at the same level within the tissue layer. If you answered yes, that your tissue is more than one cell layer, then you are looking at a stratified epithelium. They too are identified by cell shape. Stratified cuboidal tissue has more than one layer of cells that are square in cross-section. This photomicrograph is of a seminiferous tubule from the testes. You can see the little spermatids in the lumen of the upper tubule. You can also see several layers of actively dividing cuboidal cells separating the spermatids from the exterior surface of the tubule. Examples of stratified squamous epithelium can be found in the vagina, at the esophageal cardiac junction, and in the epidermis of the skin. This is a photomicrograph of the skin. The unlabeled black arrow is pointing to the basement membrane of the epidermis. Transitional epithelium is found in the bladder, ureters, and urethra. The black arrow tip is pointing toward the basement membrane. Apical cells in this tissue will appear rounded. Stratified columnar epithelium is a very unusual tissue type. It is found in the ducts of some glands and in the male urethra. In this image, you can see the nuclei are at various levels. Unfortunately, the cell membranes representing the cell boundaries are not visible. This concludes this presentation on epithelial tissue.